There's another one that's from the closing players. It's CCM. Oh, I was going to say there's too many. I mean, because on here you had a string of there's yes. Loyal. Yeah. I've never replied. That's I understand. It's kind of showing right here film, right? Right. Just I'm ready.
I'm not entirely sure why it's echoing.
You get echo? Yeah.
All right, we're trying to solve the echo issue. I mean, we could either mute this or mute that.
Hello? No. Hello? No, it's still coming through here. Is there? Can you just mute speakers on the laptop? I do have it, but I don't know why it's still echoing. I'm not sure.
Hello?
Yeah, so I have it completely muted on here. It is.
This and the bottom sometimes. Sound recorded. I think we want more. Hello?
Got it.
Yeah.
Are you guys on line hearing a hearing echo?
I have it muted online.
Now we don't, Buffer.
Okay, but you can hear me.
Yes, it sounds good right now.
All right, I'm gonna go ahead and introduce Terry and see if we can suffer here with the echo. All right, delighted today to introduce a good friend who, Terry, I won't embarrass you by saying how many years we've known each other. I think he colors his hair and I don't, but you know, whatever. I've known Terry at least since the University of Chicago days, where he trained with Gene Chang and Jeff Bluestone. Everybody knows Gene, but Jeff is a world-renowned gamma delta T cell basic immunologist. So Terry was originally studied at Notre Dame and then MD at University of Illinois, Chicago. Then for fellowship, clinical and basic science at University of Chicago, and then took up, he was a active faculty member at Northwestern before going to be division chief at University of Kentucky. So Terry is an ultimate physician scientist. He's an active IBD clinician, runs a division, has at least 2 R01 and a VA grants, runs an active fellowship program, both research and training and clinical, and has really done exceptionally well. He's a very bright guy, as you might guess from his background, and is going to talk today about an area that is tremendously exciting and kind of blends his background of immunology and epithelial cell biology with Gene. And so he's going to tell us about a completely novel area of how to reverse the abnormalities of mitochondrial function in IBD, which is a area that is just proliferating immensely. And I think is probably the answer to ulcerative colitis, which is proposed as a nutritional deficiency disease back in the, gosh, late 80s. Yeah, that was, it's a starved gut hypothesis. So anyway, Terry, thank you for visiting. We've enjoyed your presence here today. Sorry it's brief, but we'll love to ask questions as you educate us about mitochondrial function.
I was thinking about back in the very first grant, I was in Jeff Bluestone's lab and went after this career development award. And I think it was between me and Catherine Nagler Anderson, who's really brilliant researcher at Harvard. And I think they only had money for one, and Balfour was head of the committee. And back in those days, it was up to the head of the committee to go out and shake the bushes and find donors, get them to loosen up a little bit with their pocketbook. And I've heard that actually Balfour did find the donor and got that little detail fulfilled. So was able to keep me and my family out of hock there for the next 3 years.
Should mention 5 kids.
Yeah.
Including triplets.
Yep.
During fellowship.
We had 5 kids in 2 years. And my wife's unbelievably talented, as you could imagine. So this is one of those really neat areas that, if you get the right training and you learn how to ask the right questions, it's pretty cool what you could discover. Set up your models the right way and then squeeze Mother Nature until she gives you the answers. There we go. Okay, I think a lot of people have seen this. I'll just add my own twist to it that IBD we know has genetic susceptibility. We know from family members, family studies that there's clearly, the genetics don't explain everything. They probably account for as high as 15, 20%. And we really think there's a lot of environmental triggers that go into it that help to alter the immune response. And where that has been more and more aimed is things like stress, things like poor diet, et cetera, that those things are altering your microbiome, and the altered microbiome is what really triggers the deleterious immune response that we see in our patients. And so this is for those that don't spend a good part of their month behind a scope, is what it looks like in Crohn's. You see a excavated ulcer there at the top. And you can discharge this patient today if they don't go to the infusions for Remicade and they come back a month later because they're getting anemic. That ulcer will be there exactly as he looks to you right now. No healing at all. So really one of the major defects is the incapacity of these ulcers to heal without therapeutic intervention. And that's true in ulcerative colitis as well as in Crohn's. Ulcerative colitis, a little bit different, obviously more confluent inflammation with intermittent ulceration. What you see on the right there is architectural distortion. And this is what I teach the fellows. The way you discriminate between IBD and infectious colitis is chronic architectural distortion. And what that is, you can see there's crypt branching going on, interestingly, in the mid crypt there. Over here, Scott. See how that kind of branches up there, not at the bottom so much. There is fissioning as well, but we were talking earlier about where these guys could be coming from. So we see goblet cell depletion. You don't see as nearly as a number of goblet cells. And then you also can actually have a crypt dropout from apoptosis, but no granulomas that you might see in Crohn's disease. So why is mucosal healing important? Now, the short of it is that when they did a deep dive, they found that patients might feel better, but a lot of times their intestines were still abnormal. They still had ulceration, still had inflammation. And once they started looking for healing of ulcers in response to drug A, B, or C, The ones that led to healing actually had decreased likelihood of a flare, decreased progression to disease complications such as strictures, and decreased need for surgery and hospitalization. So the mucosal healing became the real gold standard of therapeutic healing. So why do they fail to heal in IBD? And I think what I really got stimulated by a bunch of studies here from Ted Denson worldwide. This is pediatric prospective trials where they biopsied new onset ulcerative colitis and Crohn's, followed the patients clinically, and then went back and reanalyzed the RNA-seq data to look and see which features of the initial presentation were prospectively linked to complications and disease. And one of the three areas that came out in these unbiased prospective trials was a deficiency in mitochondrial transcripts. So either mitochondrial biogenesis or mitochondrial function, et cetera. So that 25% of their patients had disease manifestations linked to this defect in mitochondrial function. And those in particular in Crohn's had increased strictures and increased surgeries. So it certainly started to point the finger in our direction. What happens when you have mitochondrial dysfunction? You have impaired barrier function, increased inflammation, increased cell death, and a reduction in mitochondrial mass and mitochondrial biogenesis, along with the reduced mitochondrial function. We're going to show some evidence for that as we go through this. And I just wanted to give people a notion. Mitochondria is not just like a static thing. It's constantly cycling. So you have fissioning, and if you get hit with ROS, that fission can turn into damage and ultimately result in mitophagy, which lowers the levels. You also have repair and increased protein synthesis of mitochondria, and those mitochondria fuse and actually is healthy for the mitochondria. And that's mitochondrial biogenesis. So this is constantly cycling. And when you're upstream of that, PGC1 alpha and NRF2 promote mitochondrial biogenesis. And then, as we'll talk later, mitochondrial function actually drive up things like NRF2. So it's constantly a cyclical reinforcing dynamics. In the case when you have unhealthy mitochondria, we have decreased ATP synthesis, decreased oxygen consumption, which is going to become a factor here later as we go on, decreased antioxidants. So mitochondria is intimately linked to the production of antioxidant enzymes. And that's really going to be important because we want to keep important cells alive. The way to do that is by making a lot of antioxidants so that you're resistant to the ROS that's, being released. Maybe because Mother Nature thinks there's an infection going on, in some cases there is, tuberculosis, all kinds of schisto, all sorts of leishmania, other sorts of infections in the gut. And she'll clean that out with the release of antioxidants, which poke holes in the membrane of bacteria. But also decreased protein synthesis, et cetera, for when you have unhealthy. So healthy mitochondria, the reverse. So they have electron transport, which is shown in the figure there. But that leads to ATP synthesis, increased mitochondrial ROS, which is a signaling molecule. So don't confuse mitochondrial ROS with the ROS you get out of macrophages and neutrophils. The reason why I say that is because mitochondrial ROS drives NRF2, and NRF2 is a major upstream regulator of antioxidant enzymes. So if you drive NRF2, you now drive the antioxidant enzyme. So healthy mitochondria have a lot of nice benefits downstream, including increased protein synthesis, which is going to be a big deal because if you want to create and maintain a barrier, you need a lot of proteins to be synthesized in order to build tight junction proteins, adherence junction, et cetera, that are all going to be important for maintaining the barrier. That comes from healthy mitochondria that are capable of making the protein that is required for barrier function. So why do ulcers fail to heal? So this comes up with a bunch of questions we're going to start asking. Do mitochondrial responses differ between normal and IBD ulcers? Okay. And we're going to examine ulcers in IBD that don't heal, but rather than just compare it to normal mucosa, we're actually going to compare it to healing ulcers in healthy controls. Okay, so the way we do that, first go out and get a grant. Next, you volunteer or you ask for volunteers that are going under colonoscopy for ulcer healing or irritable bowel or whatever, and you ask them, we'd like to do 20 extra biopsies and then bring you back 4 to 5 days later and re-biopsy those biopsy sites. So we call this the biopsy of the biopsy. We're going to compare the biopsy to biopsy, which is actually where we get to see what normal ulcer healing looks like. So these are, every biopsy site creates a little ulcer. If you catch it in 4 or 5 days, you can catch it in the process of ulcer healing. If you wait too long, it'll be closed up and you'll miss it. So that's by 7 days, you missed it. But 4 or 5, you catch it right in the sweet spot. All right, so that's normal versus ulcerative colitis. You can see the ulcerations here, and then here's one deeper ulceration. So this would be a Mayo 2. We typically are going to study mostly Mayo 1. On the left is immediately after I did the biopsies. So we did 20 biopsies. So we separated them by about 2 centimeters so that we hopefully decrease some of the nearby side effects. And there's a nice ulcer at around 4 days where you should see the depressed center there. There's like a excavated ulcer base. It's at this point, it's probably about a monolayer thick with some exudate, such as neutrophils, et cetera. And you've got the heaped up margins because the edges are starting to undergo crypt proliferation. And then in a normal person actually undergo crypt visioning to heal and repair that surface. A day later, you can see how fast this is healing, right? A day later, you've now started to form that layer even better. You're starting to form the crypts are starting to spread across the ulcer. And then by day 7 and 8, that has all been cleaned up. So we're going to say, okay, you need some kind of inflammation. I mean, you just saw, a picture with exudate, so there's neutrophils there, obviously. But has it been fast enough to bring in a bunch of activated macrophages and T cells and create all this damage with ROS like we see in IBD? Not really clear. We're going to find out here. So what you do here is we're going to compare control, which is, albeit, it's flat mucosa, nobody damaged it. And then we're going to compare that to mild ulcerative colitis, and then to the biopsy on the right. So some of the cytokines are increased in ulcerative colitis, like IL-1 beta, IL-6. These are commonly think of as type 1 or macrophage-inducing inflammatory reactions. Interestingly, there are some cytokines, mild inflammatory responses that are linked to the biopsy. So if Mother Nature has her druthers, this is how she'd like to heal this thing. IL-18, IL-22 being the classic ones where we actually have RNA-seq, bulk RNA-seq that's been just sent back to our lab, so they're not developed in time for this talk. Interestingly, some of these are seen in both. So TGF-beta, we're just actually trying to douse the flames. TGF-beta is regulatory. It also can cause fibrosis, which is good because Mother Nature wants to form a Band-Aid on that surface so that the bugs don't get in. Too much TGF-beta persistent will cause fibrosis. So she's got to know when to use enough and then when to stop it. NLRP3 and LRP6 are markers of different inflammasomes, as is INOS. And you see that there's different patterns that are going to start to emerge here between different inflammasome pathways that will be linked to it. So summary of this first part, UC tissue displays a robust cell-mediated immune response. We also saw TNF and interferon in this. And inflammasome activation, typical of the NLRP6 and INOS. In the biopsy, different type 1, type 17 responses, IL-18 and IL-22, which actually this group here in University of North Carolina has become interested in. So we had a lot of fun talking about potential collaborations on that. NLRP3 inflammasome responses seen in host defenses to intracellular infections were seen here in these biopsy responses. So ulcer healing in these biopsy people increases mitochondrial levels and plateau. That's our second hypothesis, because we think the world revolves around mitochondria, of course, and we think that that's one of the reasons why you can't heal. So we would hypothesize that if you let these things operate without a disease like ulcerative colitis, they should increase their mitochondria. Mitochondria should be a central player in healing the ulcer. So this is actually staining for mitochondrial complex 1. On the left, you see normal levels of mitochondria. In the middle, there's just nothing. I mean, ulcerative colitis is very low levels. ROS induces a lot of the damage, increases mitophagy, and really depletes the mitochondria for a number of reasons. One is the mitophagy I just mentioned, but also reduced mitochondrial biogenesis. And then also reduced antioxidant enzymes. So the antioxidant enzymes are being overwhelmed, which only opens the door for more ROS-induced damage. The far right is now our biopsy, the biopsy. And you can see here at the surface, normal colon has a normal level. You know, you have good barrier most of the time. Ulcerative colitis, very low levels. So that's actually really new data, cool data out of Sean Colgan's group that shows that oxidative stress actually causes degradation of E-cadherin as well as claudin. So there's clear mechanisms between the mitochondria, ROS, and damage to the barrier proteins. Biopsy, the biopsy, see, she's lining things up. She's got the mitochondria levels. They're like a little palisade, a fence up there. And that we would predict that is being used by Mother Nature to recreate a barrier in response to these ulcers. This was super cool when we saw this data. This is normal on the left. You see, again, no mitochondria in the middle, and then really a clear increase in mitochondria in the crypt bases of the normal biopsy, the biopsy healing. So if we do some quantitation, we see that biopsy actually increases over control levels as in the plateau as well as the middle crypt and crypt base. And if we go and look by message RNA, we see markers of biogenesis. So PGC1 alpha is the master regulator of mitochondrial biogenesis, as is TFAM. The mitochondrial complex proteins 4, 6, COX5B, which is complex 5, as well as AFP or ATP 5E. So you see, it's not just the complex one, but all of them, as you might expect. And keep on harping at it because I think this is the reason why this system was so interesting. A clear induction of antioxidant enzymes. So increase in the antioxidant enzyme most responsible for cleaning up mitochondrial ROS, which is good, right? Because we just mentioned if you have too much ROS around, you're going to get mitophagy, you're going to reduce the levels of mitochondrial function and mitochondrial biogenesis, whereas increased antioxidants in the mitochondria and cytosol will help preserve normal functioning and actually get the cell ready to heal an ulcer. Right? So this is how we did some staining for proliferation markers. You see on the left, normal levels of control. It's in the lower part of the transit amplifying zone with low levels in the crypt base. And now you see this expanded transit amplifying zone as you see in mild ulcerative colitis. So that's a different population of cells. Some of the ones at the bottom of that are proliferative progenitor cells, and that population is increasing. Now, why is that? This may be right next door to one of those ulcers, and Mother Nature is going to have to spread a monolayer across that surface and then begin to feed cells into crypts that are being newly generated. So she needs more proliferative capacity. Unfortunately, we talk later on the question and answer why that doesn't really quite work so well for her, but that's what she's trying to do with that expanded TA zone. Biopsy, the biopsy, also we see an increase transit amplifying, but really what's most impressive here is the crypt base. Note over here, we don't have a lot of proliferation at the crypt base, you'd almost think that should be what she's trying to do because that's where the stem cell zone is. We should be seeing stem cells starting to expand. Clearly that's not happening, whereas we do see it happen in the biopsy. So this all links together. You know, there's some papers that in your own line of research that really, really hit you and made an impact. This one was one of mine, Rodriguez-Coleman Nature paper. What she showed was that you need cells to be able to give substrate that feed oxidative phosphorylation into your precursor stem cells that cause them to become activated stem cells. In this case, it was lactic acid made by Paneth cells in the small bowel. And this has actually not been shown yet in the colon. So messenger RNA, you know, these are a little loosey-goosey down, and when you do a lot of single cell, you find out there's a lot of messenger RNA for all sorts of genes that are being made that ultimately maybe don't translate all the way out to a protein. But you see here, LGR5, AXIN2, ASCL2, and specifically olfactamidin-4. And there's also little hints as you go into an area. This is going to be some kind of secret. We haven't figured it out yet, but this is a secret as to why is the biopsy is doing so well? Why is it healing so fast, whereas the UC is not? So what is it about the sulfactometed or induction that is going to become so important. Similarly, let's look down the road, Wnt target genes. So Wnt is how you regulate stem cell pluripotence as well as expansion. So cyclin D1 is a proliferative marker. c-Myc, interestingly, a Wnt target gene, but also a major upstream regulator of mitochondrial biogenesis. So that's perfect. You want to make more mitochondria to get your cells revved up, and you need more mitochondria to be able to do that, and that's what you see here. SOX9, which is really was interesting to us, is something that we've seen associated with crypt fissioning. So if you have let's say an ulcer surface that's denuded, you first put across a monolayer, and then you actually take crypts and you start expanding crypts across the surface, and you do that with crypt fissioning. So SOX9 is an important regulator, and that also is upstream of the olfactometin that we talked about earlier. So this is starting to line up, and what really gets you revved up is when you start seeing things that are in the biopsy, but not in UC. So for example, let's say we didn't do any of this biopsy stuff. You would never know that this is what Mother Nature really wanted. You would just know these two things, right? Not much cyclin D1, not much c-Myc. I guess stem cells just aren't important because you don't get but now with this new data, you say, no, this is what Mother Nature, this is a proper control. See what I'm saying? You now know that you have a deficient response in the UC because you have the proper control, which is normal ulcer healing. ATO1, which is kind of cool because that's an innate signaling pathway, so that drives goblet cells. And remember I mentioned that in UC, one of the pathognomonic features pathologically is goblet cell depletion. So ATO1 is pushing more goblet cells, again, telling us that, UC, this is one of its broken wings. It's not doing what Mother Nature would really like to do, and that is to drive up more and more goblet cells. So it's kind of a really fascinating part of the story. So in summary here, we have this NDU-FB6, which is complex 1, increasing on the plateau as well as in the crypt base. We think at the plateau, it's helping reestablish barrier integrity, and on the base, we think it's driving up the mitochondrial levels, which are important for stem cell activation and stem cell expansion. So hypothesis number 3, enhanced mitochondrial function is going to accelerate epithelial repair and reverse colitis. So we fortunately met a guy in the University of Kentucky. We met at church under the cross, so to speak. We both had grants that were submitted. My grant was on why you can't heal ulcers and because of mitochondrial deficiency. He had just sent in a grant on a new drug he developed that increased mitochondrial function. So we're looking at each other and where we were, and that was the date of our invention. And so you see the patents that has come out of it. This is a drug that's a gold. He came out of the guy who made cisplatinum at MIT. So it's a metal chemistry lab. So gold in the middle, surrounded by these phosphine rings, which actually make it permeabilize, and the slightly cationic charge attracts it to the mitochondria. He recently has shown that it binds to VDAC, which is an outer mitochondrial membrane pore, which is a channel for calcium as well as a number of metabolites, and has been involved with mitochondrial biogenesis in the past as well as function. And this paper's finally coming out very soon, the American Chemistry Society. So now with this in hand, we can actually start talking to journals about publishing what I'm going to show you. If we give it, we localize it to mitochondria. If we feed cells with it, they increase mitochondrial biogenesis, as well as this. Remember, I told you this NRF2 is upstream of all these antioxidant responses. We could stain cells with a very specific mitotracker, which tells you quantitatively how much mitochondria is in the cell. So you can see with gold phos, we have an increase in mitochondrial mass. And I think this last figure of all of it is probably the most important, because this is where we fed mice the drug. And 3 days later, isolated the epithelial cells out of the colon and put them through Seahorse, which is a device to measure oxygen consumption rate. So this is a direct marker of oxidative phosphorylation. We could show that there's a 30% increase in OCR and epithelial cells in a gold-phosphate mouse. So the drug got into the colon, went through the mucus bilayer, into the cell, localized to the mitochondria, and jacked up its function by 30%. So to me, this is a really important figure. This is just, cool stuff for our, biochemistry nerds in the group here, just literally looking at mitochondrial protein levels. And I know I've been giving you a lot of immunohisto, but there is Western blots behind all this immunohisto stuff I've been showing you, where you could show all the complexes in the mitochondria chain. You could also begin to start localize some of the effects. So this is where we take a normal colon, give it gold phos for 3 days, and we can see an increase in the middle of the crypt as well as the upper. Remember, I said the upper was really important because of the barrier function. Again, this mitotracker green, which is a quantitative readout of specifically flow identified epithelial cells. And so that's more of a real quantitative assay. Just in advance notice, we're going to talk about 2 different colitis models here. One of them is DSS colitis, which is where you put DSS in the water. It really is something, it's like a soap. It doesn't get ingested, doesn't get cleared very well by the macrophages, who also get really angry and they get activated. And so it drives up type 1 macrophage responses and induces colitis. So it's ulceration, which is very severe in the beginning, very type 1 driven inflammation, actually not as much T cell, but then you stop the DSS and you get ulcer healing. So there's an ulcer repair and then residual levels of inflammation. What we did in this study is we gave gold phos along the colitis induction. In the second study, we have IL-10 knockouts, which we induce with piroxicam, stop at day 14, and then wait to day 28, which is really when the severe transmural chronic inflammation occurs. And that's when we start giving the drug. So we don't, in this second set of data, we'll give the drug after we've gotten severe colitis, so a true treatment protocol. Actually, we've had a couple gastro papers previously about how this model, when given long enough, can actually induce really nice colitis-induced cancer. And a paper by Gu Li was in my lab at the time. So this is where we start to have fun with the metabolomics. So this is your DSS, and it's pretty standard stuff. You get you block the colon shortening that occurs, so you could get longer colons that survive, and actually get reduction in disease activity indices, which is bleeding as well as loose stools. You get a reduction in the ulceration. So this, if you look at it, the colitis score, which is intimately linked to the amount of ulcerated surface, actually improves in that ulcer healing phase. So you have the initial 7 days, which is ulcerated in both, stop the DSS, and this gold phos actually accelerates ulcer healing. And then fitsidextran, which tells you barrier. So we're able to actually restore barrier function quicker in the gold phosphate mice. Interestingly, the cytokines go down as you might expect, but this later on, this is like day 21, you still see the mitochondria levels down, and with gold phos, the mitochondrial levels are increased. So it does work again in the epithelial cells to increase it. It also works, by the way, in immune cell populations, and we're just really now starting to go into that area, which I could talk about in the discussion if you'd like. So this is just flipping the table a little bit here. This is the IL-10 knockout mouse model, where we show the increased mitochondria as well as a reduction in a lot of those different cytokines, which are, a lot of them are, again, are the type 1 cytokines. Next, we're going to show you actual human. So what's cool is that we take the biopsies out of patients, and they're shown here, Mayo scores 1 and 2, which is 2 is getting to be a little ulcerated. 1 is really just mild erythema, maybe a little bit of bleeding. We're going to take the biopsies, run them over to the lab within about 10, 15 minutes, and then put them in a well in the cold room for 3 hours. So this is going to be with and without drug. And so this moves really fast. So you see mitochondrial levels go up again within 3 hours. So remember, it's not that I've asked the cell to make new mitochondria. I'm actually reducing the level of destruction while I'm increasing biogenesis. So the levels respond really quick because it's such a dynamic process that what you're doing is you're also reducing degradation. And that's one of your mechanisms for increasing levels. Mitochondrial copy number go up in the epithelial cells, as does mitochondrial mass. So we were able to get in there and increase mitochondrial levels within epithelial cells in human colitis tissue and epithelial cells. Within 3 hours, again, increase in antioxidant enzymes, mitochondrial as well as cytosolic, and we can see a reduction in the cytokines in panel C, which are seen, again, this is happening within 3 hours in a dish, in a well. I love this one on the bottom because this is 8-hydroxyguanosine. This is the outcome of oxidative stress on proteins. So you can see all the stress with vehicle delivered colitis tissue, whereas on the right you see Goldfuss is able to reduce this significantly within 3 hours. So because it's increasing the antioxidant enzymes, it's able to actually reduce oxidative stress rapidly in these tissues. So what do we got so far? Well, we got increased mitochondrial mass. I showed you that quantitative. Increased mitochondrial biogenesis with some markers like TFAM and PGC1 alpha. Reduction in inflammatory cytokines. Antioxidant enzymes go up. Barrier function improves, at least in the mouse models. Oxidant injury throughout the intestine goes down, and mucosal repair is accelerated. So a lot of times when you talk to folks, like Balfour, about what's the real cause of how this happens, why does my child have this horrible disease, and why is he getting sick, and why can't he go to his graduation? Or like I've had to say, probably more than I'd like to remember, where I have to blast somebody with steroids just to get them down the wedding aisle because it loves stress, right? So this is really stressful to get married. And I've had many patients where I've had to give them 40-day prednisone just so they can walk down the aisle because this is what happens. And the parents want to know why, what's going on? Well, a big reason is that there's a dysbiosis which occurs. So we're going to take a little bit of a different slant on it. So this is work done by Andreas Baumler, who I started becoming aware of about 6 or 7 years ago. And one of the points that he made was that under really healthy conditions, you really have very, very low oxygen levels in the gut. And that's been very well known. Severe hypoxia promotes obligate anaerobes, and those are good. Things like firmicutes and Bacteroides. These are bugs that make short-chain fatty acids, which actually feed they're 70% of the energy substrate for epithelial cells. So that's healthy. And when you have too much oxygen getting into the microbiome, you actually expand facultative anaerobes. And that's not a good thing. So now you've got things like E. coli, Proteobacter, Pseudomonas, and they damage the barrier proteins that we've been talking about. They actually promote inflammation to a lot of the molecules that get across the barrier. So this is the enemy. On the left is our rescue team. And what Andreas's point is that if you limit the oxygen that gets into the microbiome, you get more of the obligate anaerobes, and they keep everything the way it should be. So just to reiterate, mitochondrial levels are high in the healthy gut. Oxygen consumption, right? You got mitochondria, you got oxidative phosphorylation, you higher OCR. So you're going to consume the oxygen before it gets into the lumen and reduce oxygen delivery. The reverse is true with inflamed gut. And if we look at the enemies, obligate anaerobes are the friends, firmicutes on the left, and a healthy gut, you have limited numbers of Proteobacter, Fusobacterium, and the other facultative anaerobes. But in the sick gut, the oxygen actually suppresses expansion and blooms of the firmicutes and Bacteroidetes, and it increases the facultative anaerobes. So impaired mitochondrial function leads, this is hypotheses at this point, leads to increased O2 availability to the microbiome and promotes facultative anaerobes. Remember, I started this by saying a real key problem is reduced mitochondria in the lining, and that was what Balfour referred to. A guy named Bill Roediger did really neat studies back in the 80s to show that we have lower levels of mitochondria. If you feed short-chain fatty acids to biopsies from patients, they don't metabolize it. utilize the short-chain fatty acid, hence the term starved gut. If we reverse that, we enhance the mitochondrial function, that actually reduces oxygen delivery to the microbiome, which maybe can help us heal this. So first of all, we give our gold phos to a normal mouse. We have a marker here for hypoxia, and actually you can see it light up on the surface of the epithelial cells. So the drug does what it's supposed to do. This is one of the things I really loved about Andreas's work. So this is where he had a pair of E. coli. The wild type uses oxygen, so it loves, when there's no mitochondria and the oxygen is just free flowing. The mutant here actually prefers to be anaerobic. So if you look at the ratio between these two, it'll tell you in a real sense, a biologically relevant readout of oxygen availability in the microbiome. So we get mice, and the cool part is that mice from Jackson Lab don't have facultative anaerobes, so they just eliminate it, they kill them. They do a little agar test. If you're positive, you're gone. So that's good because now the niche for the E. coli is available. So they give this E. coli pair to the mice, which we had to buy from Jackson. And you can see the ratio of the E. coli. So in the gold phos here, you're actually getting an expansion of the mutant, right? Because they prefer the anaerobic, and you don't get a lot of oxygen because you're using it in the lining, and the ratio reduces in gold phos treated. So this is actually a biologic readout that you're reducing the amount of oxygen availability in a gold phos fed mouse. And this is actually DSS colitis, I should mention. All right, so let's start looking at the bacteria. So Proteobacter can, as you increase the dosage, just as a wild type mouse, because one of the criticisms is that our drug heals the colitis. Maybe that's why the anaerobes and the facultative anaerobes switch. This is without any inflammation. We can shift the microbiome in a normal uninflamed mouse. And on the right shows you at the genus level an expansion of the obligate anaerobes. Now this is in this DSS colitis. Remember at the end of day 21, the first 7 days, it's just a severe ulceration. It gets to heal over the next 7 days, and then day 21 is usually residual inflammation. And by then you start to see changes here, increase in the Proteobacter and in the control and in the reduction in that in the gold phos treated. And again, the relative genus abundance, so you could expand the obligate anaerobes in the gold phos treated mice at the genus level. And this is where we started to get into, I think, a model which was, just a godsend. I have to give credit here to Simon Gray, who showed this very nicely in a germ-free IL-10 knockout that was given stool from Crohn's disease patients. I think 3 patients is what I usually tell people. And you mix it together, and it's pretty neat. You get the mice that are shipped to us germ-free from here, and then we also get the stool. We get them together at Kentucky. I think we leave them get re-equilibrated a little bit from, and then give them the human stool, and they get severe, moderately severe colitis, and it has, which is typically assayed by looking at fecal lipocalin, so you don't have to sacrifice the mice. You kind of get to know who's got the active severe colitis. In this case here, we're going to give them gold phos after they get severe, so this is going to be around day 28. So again, this is a true therapeutic model. If we look in here, these are the mitochondrial stains, which you've become familiar with now. So if we give these guys, if we don't give them anything, there's low levels of mitochondria. If we give them gold phos, we drive up the mitochondria. And to be honest, this is epithelial cell as well as in the lamina propria, and we can quantitate that. When we look at the fecal lipocalin, we specifically choose the ones who have the most severe fecal lipocalin levels. So let's say if we got 10 mice, we'll pick the top 6 fecal lipocalin, and then we'll divide those between control and gold phos. So we make sure that we're comparing apples to apples. And then when we start looking at the bugs, we can see here Proteobacters in the blue, and we can see a reduction in the Proteobacter bacteria and an increase. Not as, it's always not so clear, but they're in this case the firmicutes, which again are the friendly guys going up in the green. And then a quantitation of the proteobacteria seen on the right. So it enhanced mitochondrial function, accelerated ulcer healing, reduces mucosal inflammation, but it also increases oxygen utilization and reduces O2 delivery, promotes a microbial shift from facultative anaerobes over to obligate anaerobes in the humanized colitis mouse model. This was kind of came to us where we said, all right, what do patients really want? Patients would love it if you could give them a expensive drug and then stop it. So they don't have to keep paying, they don't have to continue the risk. So we said, all right, let's see. We stopped the drug at day 57, let them go for another 30 days, and then tested them to see. And in fact, fecal lipocalin levels stayed down. So the colitis did not come back when we withdrew therapy. So it suggests that, in fact, once you fix the microbiome, that actually can act as a therapeutic effect throughout the period of remission. So this 3-month period, or 30 days, I'm sorry, 30 days. If we do a KEG analysis, we can see that tight junction proteins levels are predicted to go up, MAP kinase signaling, including PI3 kinase signaling. So these are all good for mucosal epithelial healing, short-chain fatty acid production, as well as reduced lipopolysaccharide biosynthesis. So this is just looking at 16S RNA, so bacteria-derived, and predicting what effects that's going to have on the mammalian cells. We look at the bacteria, and this is at the end. So day 58 is that period of intense colitis where we've treated, you compare vehicle to goldfuss. And so the orange guys are the proteobacteria. You can see here that they're fluctuating but positive in the vehicle, but significantly reduced in the goldfuss treated. Whereas in the red are the firmicutes. And I want to point out that sometimes you'll have a mixture of these bugs, and not all firmicutes are going to behave the same. Not all are good, and just like not all the proteobacteria are bad. So what you really want to get into eventually is function and metabolites, because those are the things that affects the mucosal level of inflammation, barrier function, all sorts of other real life functional consequences. Remission proteobacter levels really get snuffed out completely. You can see that's the orange guy here, and we're just some of these remission guys. So actually the remission microbiome looks better than the one at the end of the colitis, right? You would think that was the worst colitis, they should have the biggest effects. Turns out if you let the mice percolate afterwards, it really becomes more stark, the differences. So this is 30 days without any drug. Previously treated with goldfuss. So that's good. Same thing. This is now day 58. Again, you're seeing multiple different sets of studies here. And so the short-chain fatty acids, they're Merbaculacea, which is the blue going up in the goldfuss treated, and the lactose. Let me see, you could say it better than me, Balfour. Lactose. Yeah, the good guys. I can see they're really, they're driving up in the goldfuss treated by the day 58. I got to get better at these bacterial names. Some guy's going to get up when I go to give this talk. He goes, you don't know anything about these bacteria. You don't even know how to pronounce their name. But now this is the part that we find really exciting. So these effects are persistent. Actually, if anything, they get better over time. So again, we're in the process of doing this for the 3rd cycle now, but there's clearly an increase in short-chain fatty acid by counting the phyla and the genus. We haven't proven yet, but we're going to today before we leave, that actually what happens to the metabolites themselves. So it corrects our hypothesis that this change in the phyla are going to result in changes in metabolomes. And if you ask a lot of the microbiology guys, they've moved beyond just collecting names of genuses and diversity and all that stuff. They want to see function. What do the changes do to the function of what's happening to the mucosa? So if you look at this, and a lot of the work has come from the group here, where changes in the metabolites like reduction in short-chain fatty acid, that's the starved gut. Remember I mentioned from Bill Roediger, an increase in secondary bile acids that clearly affects the epithelial barrier integrity, decreased energy metabolism, decreased protein synthesis, as well as the amino acid metabolism. So these are the downstream negative effects that we see in IBD and how they're linked to the functional consequences I've listed. So now metabolomic analysis, we see an increase in short-chain fatty acid synthesis pathways. So we can clearly say that all of these different short-chain fatty acid pathway downstream and intermediaries are increased, as well as the secondary bile acid metabolites shown in the upper right. So this is a real big story that has been tested in multiple different groups around the world. And we can drive this and get these metabolites to change in a way that would promote a healthy environment for the mucosa. Tryptophan metabolism, so you want to get into indole, that actually reduces the inflammatory responses, and the polyamines and amino acid metabolites show that there's an increase in protein synthesis as well going on. So unlike what you see in IBD, you see a reversal and you see an increase in tryptophan. This gets a little dicey. So some of these, like for example, the creatine is you could say is mammalian cells, and you see that's going up. Creatine is important because the levels of creatine alone, not phosphorylate, but creatine alone, is linked to ATP. The creatine phosphate gets dephosphorylated, and you come up with free creatine, and that's what we see here is a significant sign that there's an increase in energy metabolism, which you'd want if you're trying to heal ulceration. The TCA cycle metabolites are increased. You can't really say that's bug or mammalian because it can happen in both. But the creatine as well as the carnitine fatty acid oxidation pathway are indicative. So what we're proposing is that we've got two different scenarios. One is healthy and one is IBD. One is the dysbiosis, which is associated with reduced mitochondria and increasing facultative anaerobes. And that reduces the short-chain fatty acids, which are needed to maintain mitochondrial function in the gut and energy substrate for the rest of the epithelial cells. With the drug, we increase mitochondrial function, which reduces the oxygen delivery to the microbiome, and that reduction in oxygen delivery impairs facultative anaerobe blooms and increases blooms of obligate anaerobes. So increased mitochondrial function increases OXPHOS, increases antioxidant enzymes, increases mitochondrial biogenesis, reduces colitis activity, and corrects dysbiosis, along with altering microbial metabolomics. The increase in short-chain fatty acid improves oxidative phosphorylation. Again, that's the energy substrate. The more times you rev up and use the mitochondria, the more oxygen you consume and the more energy you produce. So that's a good thing if you're constantly reproducing the lining of your gut every 3 to 5 days, which we know is what happens. We increase the deconjugation of primary bile acids into secondary bile acids, which is a healthy thing to maintain gut and barrier integrity, as well as correct immune dysfunction. It increases tryptophan metabolism, increases polyamine synthesis. Remember, I told you that was linked to protein synthesis, and increases host energy metabolism. So what would you like to do with this drug? Ideally, you'd like to give it to patients, or at least something that does the same thing as Goldfoss. Whether it's going to be this Goldfoss, or we're going to have to alter it to keep it in the gut alone. There's a company out of Philadelphia that's actually been able to add a side chain that keeps it in the gut completely. They've done it with their drug. We're going to ask them to do it with our drug. But the point is that this oral therapy might be capable of getting people to maybe it could be a salvage therapy in patients with really severe colitis to get them out of the hospital and get them, transition to a different drug. Potentially, because of that remission data, maybe this is something where if you fix the microbiome, you could get some people to go into drug-free remission. And again, you don't dream big, you don't get it. So that's what our idea here is: reduce remission in mild, improve remission in biologic agents, generates healthy microbiome. And it may be that the use of this drug could be beyond IBD. It could be, there's a lot of the dysbiosis has been linked to neurodegenerative diseases like Alzheimer's, the kind of metabolic dysfunction goes on with the metabolic syndrome, fatty liver, cancer has been linked to dysbiosis. So if this drug can fix microbiome and turn everybody into, a vegetarian yoga instructor, that could be a good thing, because not all of us do yoga or eat vegetables that much. And this is the lab and collaborators. And I just wanted to make a point. This just, as you can see from the team here, this is Mariana Binla. She's now got her own lab at Vanderbilt. She came out of Andreas Baumler's lab. Ashfaq Alam was actually the heavyweight that we recruited to help us with, the microbial analysis. I don't know if you remember him. He came out of Andy Nish's lab. He had a really nice Nature microbiology paper before coming. So he's been absolutely essential. Sam Awa was the guy I met in church that day. He was the chemist that trained at MIT that developed the drug. Gu Li is the guy that was in my lab and then actually he's now associate professor at UAB in human and mouse surgical pathology. So he's been actually critical to analyzing our labs. We have people here, Tatiana is from Russia, India, Pakistan, Middle Eastern. And the reason I'm saying this is because this is who we are. This is science. These are the people that are willing to sacrifice their evenings, their weekends, spend a lot of time in my, back area where we write papers and write grants and everything. And I just put this out there for everybody because we're so grateful for what happens in this country. And I hope it can continue to happen so that we can continue to come up with really cool stories like this. So thank you very much. I'd be really happy to have any questions.
So, Terry, first of all, thank you for coming. This is absolutely stupendous work that I predict is going to make a major impact on how to do treatment. So that statement leads to my question. You talked a lot about UC and the energy deficiency of UC, which I love as an explanation. What about Crohn's? Do you think this is going to have a role in Crohn's disease?
Yeah, I mean, the reason we chose UC was we had been doing a lot of work on our first paper on is about mitochondrial deficiency, and just this simple matter of doing Western blots, and it's a lot harder to get data out of Crohn's patients because the area, if you're a little bit close to the ulcer, you're going to get one kind of finding. The area a little bit away from the ulcer, you're going to get slightly different. So it's just a lot harder, and we only get it. I mean, we do this a lot. We do 20 biopsies, but you got to isolate the epithelial cells. And you know as well as I do that your error bars mean it makes something, so I think it was a lot easier to do UC, more consistent. But I think that everything that we see here is going to be applicable to Crohn's. We've done some of the cultures in vitro, and we see a very similar pattern with Crohn's with, again, more variability. For your first set of papers, you usually pick the low-hanging fruit, right? But I do think that, because I actually think that the mitochondrial dysfunction is also predictive of fibrosis. And we've seen that the worst mitochondrial deficiencies have been seen in areas of fibrosis where strictures are. So it'd be really cool to be able to start assessing those questions, with the more money that we might get. Others? Yes, sir.
Yeah, great, excellent set of work. I have two questions. So the lost mitochondria, as you showed, is both in epithelial cells and in leaf cells. Yes. There is perhaps an approach with transgenic overexpression, cell type specific transgenes for BDAT1, try and restore mitochondria in one component or the other, take that apart. And second question was, there are inhibitors of mitophagy, I think, published in trials for ALS, as we mentioned, cardiac failure. So would that be sufficient to enable?
Yeah, there's that, and there's also trials that are in phase 2 already for the antioxidant enzymes. They haven't been doing as well. We haven't had much news out of them, and I think the reason is because it's not just going to be one thing. I think if you could promote mitochondrial function, you do a lot of things. You rev up the mitochondrial biogenesis. You also rev up the antioxidant. And you're revving up the antioxidant at the place where you need it, in the mitochondria, right? In the cytosol. When you're given an exogenous agent, you're hoping that that agent gets into the cell that you want and gets to the right place in that cell and stops up. And ROS, as I'm sure you know, boom, it works. I mean, milliseconds, right? And which is, I think, why we see the effect that we do in the wells in 3 hours. So I think the fact that we're inducing antioxidant enzymes at the site of damage is why it works. And I don't think there's studies which you might say is good or bad, but I think it's actually good for us because I think ours is going to be more potent. I think NERV2, everything we've ever looked at, NERV2 has been a really reliable signal, and that's major upstream regulation. It's actually more of a in cancer than in IBD, but it's you know, hopefully we'll show that it's a very important factor in this drug effects. The VDAC, I didn't think of that. You're saying like overexpress it and see if that'll help. Yeah, that's not a bad idea.
It can be done in a cell-specific manner.
Right.
With these hardwood cells.
And I think it'd be really cool to, and we've actually, in this paper that's going to be coming out, it shows like if you alter some of the nucleotides in VDAC, you reduce the effect of it, you know, so you can, so that there's actually data, proteomics data that shows where in the VDAC molecule. I have a sneaky suspicion that some big fancy drug company is going to look at this paper and say, thank you very much. We will make a drug that is right where you want it to be, but ours is going to be really safe and it's not going to be metal. And thank you again. Goodbye. I mean, I suspect I don't care. I mean, I really think I would like this story, you know, to get into this. That'd be a great finish to my career, you know. All those tens of thousands of dollars that you and I did earn because we were at scoping people out in the suburbs. It'd be nice to say, you know what, grandkids, we gave that drug to help, you know, grandma prevent, you know, Alzheimer's, something like that. That'd be fine. That would be, I'd be happy to go into the memories of all the people here, you know. I remember meeting that guy. That'd be good for me. Yes.
That was really nice. So I'm sort of interested in why people get ulcerative colitis. And so you've shown that the drug, the mitochondria screwed up and healing. Have you biopsied UC in remission to see if the mucosa is fundamentally screwed up before they get it?
So the answer is yes. And the answer is your mitochondria go back to normal. We haven't figured the trigger. The trigger is still, I think, bacterial fighting out, and we still have. I think diet is going to be relevant. I also think that ultimately epigenetic changes that occur in the epithelial lining and in mucosal immune cell populations, I think, are the reasons why our patients look fine and feel good for a while. You know, because there's always patients who quit their therapy and all that. And the data says that, yeah, at least 50% of them will recur. If you take them off of therapy, they're going to recur. I think the reason they recur is because of the changes that are reduced in the stem cells. I think typically stem cell populations have long-lived resident immune cells, and those epigenetic changes are what drives. Again, that we're excited about that remission data, but you know, 30 days is cool. It would have been. I mean, it could have been better. It was really pretty neat. We don't know what it means for you know, if we reverse the dysbiosis now, will it give us another, you know, 3 years of remission? I don't know. But if they're going to be fighting that out, you know, that'd be fantastic for me. It means that we would have made it. We would have made it in humans. You know? And the only place we're really going to care about is what this does.
I have one more. There's some genetic conditions where your mitochondria is screwed up. Do those people, are they more likely to get colitis? And then there's drugs that cause mitochondrial dysfunction, like NSAIDs and statins. Are those associated with colitis?
Yeah. So actually, in NSAIDs, you can actually get fecal epithelial and fecal calprotectin levels up. And if you can get diarrhea, you get ulcers. And actually, so you have to be cognizant of when I do fecal calprotectin as a rule out, right? So if you come in with diarrhea, the first test I do is fecal calprotectin, because if you're normal, odds are you don't have IBD. If you're elevated, I then ask you, do you take NSAIDs? If you're taking a lot of NSAIDs, that can elevate fecal calcium and give you diarrhea. So I think the other question is,
Neelix,
huh?
like the genetic condition.
Yeah, so I've looked for that, and it's not all of them, but I'd say probably about like 40 or 50% of the mitochondrial dysfunction also have diarrhea. They've got a lot of other problems. So you can imagine muscle strength and all the other stuff, paralysis, abnormal, abnormal cardiac, abnormal mental development as well. So maybe this could help them, especially if we get a safer version. I mean, I don't think this guy. We haven't done anything yet to keep him in the gut alone. I think that's our next move. We're doing the toxicity. So far, there's no toxicity big signal that we've had. But we have to do a lot more to be, even for getting the phase 1. I'd really like to see, in vitro cultures and make sure that I don't screw up the myocytes and brain function. Well, I mean, you can only get as far as if one grant only gets you so far down the road. You need to be able to, and you need to get other people, involved with our research. This is the only group that's we want this to. So Sam's going to have to make the drug in higher quantities, especially for the phase 1 and even to get an IND, he's going to have to make all this GMP material to be able to do all the scientific studies, same stuff that he makes for humans, right? Which is already done, perfectly sterile. So millions and millions of dollars. They say now that I spent my time in Janssen and Lilly, they moved drugs all the way. It's taken $1 billion for Rizzi, for some big blockbusters that you see advertised on Super Bowl. Get any one of those is just it's there's a lot of money in. You can imagine there's a lot of money in this. So each one of those is making 3 to 4 billion. So they're putting those ads on Super Bowl for a reason, because they're making a lot of cash. That's global. Before Humira went out, I think it was higher.
Yeah, well, I think in the interest of dinner, we need to probably, thank you so much.
Yeah, really enjoyed this
stuff. Absolutely fantastic. Now, I want to. Order population.
Thank you, everybody, for showing up online.